All right. Welcome to our second take of the Network Science Introduction class. My name is Jacob Cram. I'm going to be telling you a little bit about how, so today we're going to be focusing on the part of network analysis where we look at the statistical association network. So there's lots of kinds of networks you can build. The network is essentially any kind of illustration of connections between lots of variables or lots of parameters. In a association network, we look for statistical associations between pairs of parameters. Let me give you an idea of what one of those could look like. So this is a paper that some of us wrote at University of Southern California to look for statistical associations between a number of microbial species level groups in the ocean. And the idea of the way a network generally works is that you can imagine that you've got a number of variables. In this case, is a time lag network. We're going to be talking about non-time lag associations in our hands-on today. The idea is you have a number of different parameters, and if those parameters are related, say in this case, you've got A and B are statistically correlated with each other, you might draw a line between A and B. And in this particular case, we said if there's a time lag association, we draw an arrow from the one that happens first to the one that happens last. If they're negative associations, you might draw a line like that. And so what you end up with are networks that can end up looking something like this. So we have all of our different parameters, and the parameters that are associated with each other are connected with some sort of line. So we're going to be talking about a couple kinds of statistical associations today, but we'll dig into that as we get to them. To follow along with this tutorial, you're going to need to get a couple of things set up on your own computer. First of all, you're going to want to navigate to this particular GitHub repository. So that's just github.com/BVCNet, which is the GitHub for this class, and then a word is called Network Science Introduction. You're going to need to have Git on your computer, and if you don't, you don't really need to have Git on your computer. You can also download this as a zip file. So if you go to the clone or download file, you can download it as a zip file, or better yet, you should install Git on your computer and then clone this particular repository onto your computer. So the way that would work is you could just clone with HTTPS, hit that little clipboard link, and you'll bring up your terminal, and you'll go to some folder where you want to download it, and you would type git clone. That's how you spell clone, and you paste that over. On a Linux, you do Control-Shift-and-V to paste, and that gives you the repository. And if I was to hit enter, it would make another copy of this on my computer, although I have already too many copies, so I won't do that right now. In addition to downloading this repository, you're going to need some software, all of which is freely available on the internet. First of all, you will need R and RStudio. And once you've installed R and RStudio, you're going to need to install a number of different packages. Most of these packages are available through CRAN. It's the Comprehensive R Archive Network, which basically means that once you have R running, if you just do install that packages and then you put the thing in parentheses, for instance, tidyverse, that will install the tidyverse package, and you can do that with these other packages as well. There might be a few more if once you start running things, R complains about you that you don't have some library installed, you can install it the same way. One package that's a little bit more tricky to install, but absolutely critical for this tutorial is one called Speakeasy. Speakeasy does a lot of the more sophisticated networks, especially ones that Jake Wiseman is going to talk about next class. But also the sparse correlation coefficient that we're going to do today, you'll need that. The way you install Speakeasy is you'll need to first load the library DevTools, which you may also need to install with install packages. And then you're going to run install GitHub with this little command here. And that basically is going to install this package directly from GitHub. It's not on the R network, but it is in ZDK123's personal or public GitHub repository. So once you have that all installed, you'll have all of this stuff in a folder on your computer, and on my computer that looks a little bit like this. So we have our Network Science Introduction R project, and if I double click on that, it will probably preload the data that I need. I'm just going to double click here and bring RStudio over into the window where you can see it. And so up front, we have our network science markdown document. If we didn't have that, if that was closed, I'm just going to close out everything. You can go over here to the lower right-hand corner. There's a little tab that says Files. And if you double click on the network science sparse.rmd thing, this will come up. And what we're looking at here is an R Markdown notebook. So the way R Markdown notebooks work is that we have some text. So I like to talk a lot or write a lot about what I'm trying to do and what I want you to take away from this. We have text up here, and then we have R code and these little code blocks. So this code block loads in all the different libraries that hopefully you've installed, and also this one little function that I sometimes like to use. So if you hit this little button in the upper right-hand corner, that library will run itself. And sometimes it'll give you a bunch of output like this. And if you look in the console, there will be some information there about what happened. And what you can do as you follow along is you can actually just run each one of these pieces bit by bit. And what we'll do is we will go through this and talk about what each thing is doing. We're going to do three kinds of things today. First, we're going to do a simple correlation network using a Spearman correlation. Unfortunately, with Spearman correlations, they are susceptible to some statistical artifacts. So we'll try to get around those with a couple of different ways. There's two big artifacts that you have to think about when you run big correlation networks. And the big one is what's called compositionality. And compositionality, most of the time, your data is going to be part of some data set that sums up to 1. So you have relative abundances. If you add all of your relative abundances together, your total is always going to be 1, regardless of what the actual abundance of that data is. That's even true if you didn't normalize them. If you just have read numbers, that compositionality problem still exists because your total reads doesn't actually tell you anything about the environment. So to get around that, we're going to do two things. We're going to do, first, we're going to do a centered log ratio transformation, which converts our data from relative abundances to ratios. And those are actually going to be ratios to basically the abundance of the average operational taxonomic unit. And then we're going to use a program that's actually designed to deal with the compositionality problem, which is called sparse CC, which is part of that Speakeasy package. And then the other statistical problem that often emerges is that when you do a network, you're doing lots and lots and lots and lots of comparisons. And when you do lots and lots of comparisons, and you would say, I only want comparisons that are, a p-value of at least 1%. That means that if you do, 1,000 comparisons and 1% of them are going to be spurious positives, that means that you're going to have a lot of false positive results. And there's ways that we can correct for that high rate of what we call false discovery. And so in this process, we'll do some false discovery rate calculations as well. So after we load our libraries, we will bring in our data. This command here just pulls data about this is data about the relative abundances of 16 S genes from a metagenome shotgun library. So this is our data. We're going to do a couple of pre-processing steps. So all of the analysis that we do requires a matrix. And for the most part, when I work with data, I like to work with the data as a data frame. So this is going to do a little bit of, it's going to clean up the data a little bit, and it's going to convert it into a matrix, and it's going to transpose the matrix so it looks like this. We've got our species on the columns and our different sample sites on the rows. And this is really all the information we're going to have. There's lots of environmental data from Terra that we're not going to be looking at today. A little bit more processing, and then we'll do our correlation analysis. So I'm using the function to do my correlation analysis. I'm doing a function called core test. And it's similar to the base correlation thing in R, except that it gets you p-values. So we run core test, and that gets us our information. And then once we have that, we do a number of processing steps on the results. And I'm not going to get into the details of how exactly all of this is working right now. But if you run this, it should clean up your data such that it gives you a nice thing that like this. We have our species, our first species, our second species. Rho is the statistic that a Spearman correlation gives you. P is the p-value for that statistic, and then FDR is our false discovery rate corrected p-value. And that's all the information. And then what we can do is we can plot the results. And somewhere in here, I wrote down where I found that there's a clever solution for plotting the results of one of these sorts of association networks. We end up, if we do these steps, and again, you're welcome to borrow any of these for any of your own analysis. It's all fairly technical, but a lot of this stuff should work if you just sort of transplant it into another environment. And you end up with a correlation matrix that looks something like this. And I wish it all fit on my screen at once. Let's try zoom out a little. So here we have all of our species, and for each pair of species, we have some sort of row score between them. So if every species to itself is a perfect correlation score of 1, and the other species tend to have, generally in this particular network, you see a high preponderance of positive correlations, positive associations between different pairs of variables. And if it's statistically significantly positive with a p-value of better than 0.01, I drew a little circle on that pair of points. So this is just a way of taking a first look at your data. So we're going to pause here. I think this is a good spot to jump to the next video. So if you have any questions about that, feel free to post them to the Slack channel. In the next section, we will talk about doing similar kinds of analyses using the CLR transform, Spearman analysis, and sparse analysis. Welcome back. Last lesson we talked about generating some association data using Spearman correlation networks using the psych program in R, using the core test function. And today we're going to talk a little bit about some other ways that we can identify associations. One limit to Spearman correlations or Pearson correlations, or really any sort of correlation network, is that they're very susceptible to a statistical artifact that people call compositionality. And there's a couple papers that I can direct you to if you really want to understand. There's a couple of good papers that will help you to understand the compositionality problem. This is one example. There's another example by Lovell et al. Just this one, which recommends a particular method to dealing with proportionality or with compositionality, although it doesn't identify negative correlations very well. The general nature of the problem is that when you have data that all sum up to 1, and then you try to correlate to each other, weird stuff can happen. So I'll give you an example. So imagine we have 3 variables, and variable 1 and 2 are not related to each other at all, and variable 1 is increasing over time, or space, or it's increasing from left to right in some transect. You could imagine that these aren't related to each other at all, but as this one increases, it looks like relative to these, 1 and 2 are both decreasing in abundance. What this means is when we look at them in terms of their correlations, 1 and 2 appear to be positively related to each other since they're both decreasing as number 3 goes up. So one way that we can deal with that is by, instead of looking at what we call relative abundance data, we can look at ratio data. And one way that we can get ratio data is using a centered log ratio transform. Let's look in the code a little bit about how we do that. So the way a centered log ratio transform works is it finds the average relative abundance in each one of your samples. So you basically find the average species. And instead of using what we call your regular arithmetic average, it uses a geometric average. I direct you to Wikipedia if you want to learn more about geometric averages. And it divides everything by that average operational taxonomic or unit or species or whatever kind of variable you happen to have. So this is our Markdown file that we were looking at last lesson. One nice thing about our Markdown notebooks is that they make these very readable HTML files, which you can just double click if you're back here. Here's the HTML file. If you double click on that, it'll open it in your web browser, so you end up with a browser that looks like this. So this was the Spearman correlation matrix that we had last time. Again, these are our different sets of species, or actually species. These are phyla level groups. These are different phyla, and the color represents the Spearman correlation between them, with blue being more positive and red being more negative, and the little dots indicate that it's statistically significantly positive after we do a false discovery rate correction. So today we're just going to start off with doing a centered log ratio transform. So this just goes through and divides everything by the geometric mean for that site. And Then all we do is we take that and we feed it exactly into the exact same analysis that we did for the Spearman association for this last figure. So we run the test, we do a bunch of processing steps, we recalculate the false discovery rate, and then we plot everything again with the exact same format, and we end up with something that looks a little bit different. So as you recall up here, we had lots of positive associations between everything, maybe two modules, like maybe these bacteria up here are more related to each other, these ones are more related to each other, and there's still some positive relationships between those. Over here, we have sort of more distinct modules. So here's sort of a group of correlating, associating bacteria. Here's another group, and they tend to be sort of negatively associated with each other. This bug over here seems to be negatively associated with all of these ones. So there's maybe a little bit more structure to the data, lots more associations that are very close to zero, not statistically significant. So in addition to doing a centered log ratio transform, just doing everything exactly the same, there's a statistic that is designed for this problem exactly. Is the recommended way of doing things, although it's a little bit more finicky to get working. And that's the sparse correlation coefficient, or sparse CC value. Sparse CC is a function that is part of that speakeasy package that you hopefully have downloaded and installed. And the way you'd run it is you'd actually, to just get the sparse CC values, this doesn't give you p-values, but you can just take the same matrix that you're using for everything else and run sparse CC, and it gives you an output, and it looks something like this. Now, you'll notice with the sparse CC, the output doesn't have row names and column names, which is kind of a pain to deal with, but you can always just take them from elsewhere and stick them back in. And so I've named this matrix. The output is this. There's actually two things in the output. There's a correlation output, and I think there's a covariance network as well. You could just load all the names right back in, and now you have them. And then we can plot the sparse CC correlation with basically exactly the same kind of way. So here we've got our different species, and we see there's maybe a little bit of a different structure with maybe a couple smaller modules of positively associated groups as opposed to two big ones and lots of negative associations between those. One thing you'll notice is that we haven't calculated the p-value yet, because that isn't just built into the basic sparse CC. The way that you calculate a p-value with sparse CC is that first you bootstrap your correlation scores. You figure out what the sort of your confidence is around those, and then you calculate the p-values from those bootstrapped estimates. And I'll show you how that works now. So you're going to run this program called sparse CC boot, and sparse CC boot takes a little bit of time to run. So I found if I do about 100 of them, it ends up taking about a minute. And so if you do 1,000, it takes about 10 minutes. You can make things happen a little bit faster if you can. This will take advantage of multiple cores, CPU cores. If you have a multi-core computer that you're working with, you can increase that number. But if not, you can also just let it run a little bit longer. And go get yourself lunch, and come back, and it'll be done. As you start getting into larger and larger data, so this is a pretty small data set. It's just phyla from Terra. Above, I didn't show where I did this, but we only took phyla that occurred at least 2 times in the that had an average occurrence of showing up at least 2 times. So as you start to have larger data sets, this bootstrapping test, all your tests, but especially this bootstrapping test, take longer and longer. So multiple CPUs is one way to deal with that. And then once you've done that, you can take your bootstrapped information and you can calculate the p-value from that. And then that will give us a data frame with our correlation scores and our p-values. Of course, the way it's structured right now, it doesn't tell you which thing is associated with species. So then we do a bunch more trying to keep track of which variables are which is this code here. You're welcome to just dig into that code and use it for any other projects. And we'll get into the details of it. And then, like we did with our other analyses, we're going to need to add p-values in. We can do that again. Or sorry, adjust for multiple comparisons like we did with the other analysis. And then we can plot the data, and we end up with the same data, right? So the same colors, but now we have circles for where we have statistically significant positive associations. And you'll notice that the things with the higher correlation scores tend to be the things with the lower the p-values, things that are more statistically significant. And CalPlot is a program that I didn't tell you to install yet, but you can install it, and it will let you plot multiple plots together. So here we have our 3 different analyses. Up here we have our Spearman correlation. Here we have our centered log ratio transform Spearman correlations, except that it didn't run on this particular version. And then down here we have our sparse CC values, and they look different from each other. And then last thing we need to do is we need to write the data out, and then we'll be able to load that into Cytoscape or any other network correlation program. And we'll Cytoscape in our next lecture, but if in your life you might want to load it into something else. And here I do a little bit more data processing, and I basically combine all the three different kinds of analyses together. Here they are all combined together, and then I write them all out into this file called Terra Oceans All Analysis. The P100 here indicates I did 100 permutations when I calculated the p-value. 100 permutations isn't really enough permutations. You probably want to do 1,000 permutations. But since I wanted this lesson to end eventually, I saved this one out now. But in that same repository, there is a 1,000 permutation version, which is going to get you a more accurate p-value. You should use that one for subsequent analysis. That's the end of this lecture. Tune in next time, and we will talk about how to get these sort of results into Cytoscape and take a look at them as networks.